Yes, ma'am. Yes, ma'am. Yes, ma'am. Good morning, one and all. So today we have assembled here for conducting this wonderful workshop on systematic review.
It's systematic review and
System-
meta-analysis.
Yes, ma'am. Just a minute. So systematic review and meta analysis. So we have our one of our speaker, Shinduja. She is currently an associate copy editor at Taylor and Francis in Bangalore, and she's also pursuing her PhD parallelly at Jain University. She has got research experience on effects of climate variability on the transmission of vector-borne livestock diseases in India. And she has held several research positions such as SRF, JRF teacher, and being a copy editor. She has got extensive experiences in writing research manuscripts, editing research articles, conducting statistical analysis in field of bioinformatics, microbiology, and molecular biology. She has got a strong background in training students and professionals in systematic review and meta analysis. She is also passionate about teaching and has been an active speaker in delivering technical presentations. So, ma'am, with this brief introduction, I welcome you for this workshop event. Over to you, ma'am.
Thank you, Ms. Indrani, and good morning, everyone. First of all, I would like to thank Ms. Anjali ma'am for giving me this opportunity to present. This is one of my first kind of presentation at BioClues, so I'm very excited as well as looking forward for today's presentation, ma'am. So I would request you all to please make the note of questions while I give the presentation, and we will have a Q&A session at the end of the presentation. And please find like in case you have any questions in between, please do enter it in the chat box so that I can address it once I complete my presentation. So can I go ahead and share my screen, please?
Sure, Shinduja. Thank you very much for obliging and conducting this workshop. The floor is yours, okay? Thank you.
Okay, ma'am. I hope my screen is visible to all of you all.
No, ma'am, it's not visible.
Not yet.
Can you not see the presentation?
No, ma'am.
Okay, just give me a minute.
Sure, sure.
Is it visible now?
Yeah. Yes, ma'am, you can make it slideshow. It will be completely. Yeah.
Right. So, a very good morning to one and all who have registered for today's webinar. So, the topic what I have chosen is an insight into systematic review and meta analysis. So, most of you would have heard regarding this particular term called systematic review and meta analysis, and you would have come across many research articles. So, before I could start, I would like to get some thoughts, or like what exactly do you people think is systematic review and meta-analysis? Can I get some answers, please? Anybody?
Could you repeat the question, please?
I just wanted to know what is your idea on systematic review and meta-analysis.
Meta-analysis, I know that it's a review where you even do some statistical analysis on the all the reviews collected.
Okay.
Whereas systematic review may not contain or may not contain this statistical analysis.
Okay,
I'm
fine.
not sure.
Yeah. That's quite a good answer. So exactly as what ma'am told, systematic review is nothing but a systematic way of reviewing our research articles for a particular study. Okay, so when, for example, if you ask any master degree student to go and collect some literature for a particular project work, so what he would do is he would type the name of the particular project, for example, DNA coding or genetic code. So he'll just randomly go and type into Google, and he'll try to get some hits, and he'll start collecting some research papers, and then he'll make it into one particular word file, and he's going to submit that. So that is nothing but just collecting a very basic literature, and this is not systematically done. So this systematic review is nothing but when you start analyzing the literatures based on your research topic, and when you narrow down to the most relevant articles in a particular manner. So there are many like for example, many checklists and many procedures which was followed in systematic reviews. Okay, so once you need to systematically review any topic or any research for that matter, you need to follow certain guidelines and checklist. Okay, so based on that you will be able to get a broader aspect or a broader outlook of your research or a research question. So and the next step of systematic review is nothing but meta-analysis, where you conduct a statistical analysis for the data collected in systematic review, and you give a numerical output for the complete set of data reviewed. So how good is your research, or can you proceed with it? What is the positive aspects and what is the negative aspects? So is it statistically significant? So this kind of analysis is done in meta-analysis. So in this presentation today, I will be briefing you all on what exactly is systematic review, and what are the different aspects, terminologies used in systematic review, and how can meta-analysis be done, and what is the softwares that we'll be using to perform meta-analysis, and different terminologies what we come across in systematic review research papers, and the different terminologies used in meta-analysis. Okay, and there are many statistical softwares that can be used to perform meta-analysis. So we will look into those kind of softwares, what are the different softwares, and we will try to work on our software in which we'll be running our codes to generate meta-analysis. So once this is done, like once we have a different kind of data, how do we interpret the results? We have some, for example, we have some plots. So how do we understand the plot? How do we interpret the plot? And how do we justify our results? So this is very important. So we will look into that. And there's something called bias assessment and publication assessment. So we will be having a general outlook on what exactly are those, and what are the different applications and limitations of meta-analysis. So
Ma'am, Shinduja, are you presenting it or is it in the first slide only?
it is just in the first slide, ma'am.
Okay, thank you.
Okay. So as I told you, I've come to the second slide and hope you are all able to see that. Yes, understanding.
No, it's still the first slide.
Uh,
No, it's not moving, ma'am. It's not moving.
it's not moving.
Yeah, maybe you can reshare the screen.
Yeah, I'll do that. I'll do that.
Yes, yes, ma'am.
Can you see it now, ma'am? The second slide, understanding the different
Not
types of
yet.
reviews.
Not
No?
yet. It's not visible. Not yet, ma'am.
Okay, maybe it's loading.
Yeah. We'll wait.
Okay, what I'll do is I'll just stop the screen share and share again.
Yes, ma'am. Yes, ma'am.
I hope it is visible now.
Yes, ma'am.
Okay, so the slide says it is understanding the different types of reviews. So reviews can be classified into three basic types. So one can be called as a very basic review, the general term which we use, as I gave you an example of, like for example, a master degree student who will be asked to review certain topic for the purpose of his project or for his assignment. So he'll just go into the Google and just type the topic and pull in some data from there and consolidate it in the form of a Word file and then submit. Okay, so that is a very primitive form of a review. The second type of review can be classified as an overview, where a review strives to comprehensively identify and track down the literature, wherein systematically we review, keeping in mind what is our target. And like for example, if we're going to look into how many infections or COVID infections are there present in India right now in Bangalore. So first thing what we will do is we'll go to the database. Like for example, it was COVID earlier. We go into the database, then we go and check what are how many males, how many females, the date of infection, and how many of them have been vaccinated, first vaccination, second vaccination. So this is a consolidated form of a systematic review wherein we analyze each and every aspect of the of a research question. Okay, so and in re- okay, fine. So we have done systematic review. Right now we have the data, for example, of that of the COVID. Okay, next what are we supposed to do? Based on this data available, I am going to study the comparison of COVID infection in different states. So I'm going to pull up all the data, make a Excel sheet or an Excel data for regarding pertaining to the number of infections in males and females, maybe for the last 1 month, tabulated in the name like males, females, the number, the date of infection, and the dosage, like the date of vaccination, have they taken the first vaccination, second vaccination. So all this will be tabulated, for example, for about 10 states. So this will be statistically analyzed, and once we do the statistical analysis using softwares, we will be able to make out of the last 1 month which are the major states which are having infection, or like is it the males or the number of females in different state are getting infected. Okay, so this is nothing but the total outcome of meta-analysis. So when you look into the image onto the right hand side, it says when you just pull in your complete literature, it is nothing but the basic review. And then when you systematically start reviewing individual articles, you will get a narrower perspective of your research question. Further down, when you conduct the statistical analysis, which is nothing but meta analysis, you will come to the final conclusion that what is the individual data and what is the outcome of this particular research question. Okay, now let's look into how systematic review works. So basically, we have a research question in mind. Okay, for example, I'm going to analyze the number of.
Ma'am, sorry to interrupt you.
Yes, ma'am. I didn't get you. Hello.
Could you just make it an full screen instead
Full
of
screen mode, ma'am.
yeah?
Hello? Hello,
Ma'am,
ma'am.
I have put it in full
Yeah, I'm
screen.
saying like it's not in full screen mode, ma'am.
Oh,
I think you need to share your whole system screen instead of just one tab. That's the problem.
okay. I'm sorry. For me, it is visible like a slideshow. Let me just do that 1 second. I'm
Yeah,
sorry.
yeah, yeah.
Yeah, yeah. Is it visible right now, ma'am?
Yeah, yes, yes. Yes, ma'am, it's perfectly fine. Yeah, you can proceed.
Okay, okay. So what I was telling was, let us try to understand the systematic review. So basically, for example, like earlier when I was working as a senior research fellow at ICR Nivedi Alanka, we used to work on disease frequency and the type of veterinary diseases which is confronted by usually the cows and the poultry animals. So like I have worked on brucellosis. like we try to pull in the data of brucellosis infection in different states of India. We give a consolidated output and try to tell what is the percentage of prevalence of brucellosis for the period of about 5 to 10 years in India. So first thing how we go about is we fix the research question. Okay, I'm going to start pulling in the data for brucellosis. So first what I would do is I would write down my I would make a fair note of, okay, fine, I would work on first the brucellosis infection in India of from the different states. Then my I would be concentrating on animals such as cow, sheep, and buffaloes. Next, I would be working on the type of infection, whether it's a primary infection or a secondary type of infection. Okay. Now what I would do is I use certain keywords. I go to PubMed or NCBI. There I give the keywords brucellosis, infection, poultry, and if not poultry, cows, buffaloes, sheep. Then I start reading through the research articles which have been published with the infection data. Okay, so that would be the next step. So I'm not narrowing it down. In that I would be getting about, suppose when I search I will be getting about 100 hits. For example, there are 100 papers reporting brucellosis infection in India. First, the first form of filtering the research articles would be to read the abstract. Once I read the abstract, I will get a fair idea of whether they have reported the number of infection, the number of positive infection out of the total number of animals which have been tested. So that is again narrowing it down. Then I will make a list of all, I will start downloading all the research papers, I will store it in a folder. Next would be data entry. So each and every study from this research paper would be entered in an Excel sheet in the form of different columns. Look, the columns will be named as author, the study name, year, the total number of events, the total number of positive events in this case. So the data, how it has been saved, and like how it is read in the software, I'll be sharing in the upcoming files. So after doing all this, I will be having I will be narrowing it down. Like for approximately, I have got 100 articles after I search in PubMed for the number of brucellosis infection in India. Once I start like once I start entering the data in the Excel sheet for analysis purpose, I will get to know that only 35 articles out of these 100 are relevant to my study. They provide the complete information which is required for my analysis. So I have narrowed it down from 100 to 35. So these 35 are nothing but getting out of the 100, they are just getting filtered, and which are used for the final meta-analysis. So basically the second image onto the right hand side says, so the data is it's extracted from the different studies. We assess the quality of the studies, and then we combine the complete data into one particular primitive data, like for example Excel sheet, and then we try to conduct the systematic review and the final meta-analysis. So it is just a narrow down procedure and selecting your best research literatures for your analysis purpose. Okay, there are certain protocols to follow for systematic reviews. So, and especially Prospero is one such protocol which is used by medical and healthcare professionals. Apart from that, there's something called PRISMA guidelines, which is to be followed for systematic review. And for any research paper that you're going to submit, along with for meta-analysis purpose, you need to submit a Prisma checklist. So this Prisma checklist was available online. I will share the link with you. That will give you the different aspects what does need to be covered in your research article, and how about going with, like, for example, abstract, and abstract what should be present, and if the number of participants, the number of outcomes, all these things would be addressed in your Prisma checklist. And I will show you the Prisma flow chart which needs to be prepared when you are conducting a systematic review. So as I told you, Prospero is a registry for medical and clinical systematic reviews. OSF pre-registrations are systematic review and protocols, including non-medical topics, which choose to affiliate with temple universities. And there is another protocol called as call share, which is again another form of review protocol. And there is systematic review protocols for animals and food, and another review protocol by Johnsburg Institute for systematic review register. Okay, so that was introduction regarding systematic review. Now let's understand what is meta-analysis. So as I can tell you, it is an extended version of systematic review. Okay, so a subset of systematic reviews, it is systematically combined, and then using the qualitative and the quantitative data from several selected studies, and then we give a final conclusion with a statistical analysis. So the numerical value what we get after performing systematic reviews is nothing but the results of meta-analysis. In systematic reviews, we only try to pull in the articles. We have the raw data with us, but how do we justify the data what we have got is true, is correct or not? Can we proceed further with the research? So this justification is done by meta-analysis. For a basic example, now like since we had the pandemic very recently, I can give you a very basic example. Once we had infection in the earlier stages, we didn't know how to test COVID. So that time, initially it was only PCR, and then after that they started developing the swab test, and then like we have lab to even using the micro array technique, we can test whether a person is having COVID or not. So these kind of tests, when they have conducted in large number of patients, the data has been pooled and into the repositories. So when we start pooling in, for example, 100 data, like 100 data of 100 patients who have been tested using PCR, and data of 100 patients who have tested using just a swab test, and like for example another 100 patients. So all this we start pooling, we do the statistical analysis, and finally we get to know, okay, the PCR is the best technique that can determine for COVID infection or not. So that can be the result which can be obtained from meta-analysis, like statistically proving your data and going forward with the research or your the outcome of your research. Okay, so this conclusion is statistically stronger than the analysis of any single study. So due to this, the increased number of subjects with greater diversity among the subject is accumulated, and the effects and the results have been justified using meta-analysis. Meta-analysis also includes a consolidated and a quantitative review of a large and often complex, sometimes apparently conflicting body of literature. Like we have, for example, now if you look into COVID data, there is n number of literatures available in net. So how do we start pooling or narrowing it down for your research purpose? So since the quantity of data is huge, systematic review plays a very important role in selecting the best data for your research, and then statistically, really sorry, statistically justifying it using your meta-analysis procedures. Okay, so why is this meta-analysis used for? It is basically used to establish statistical significance with the studies that have conflicting results. For example, we have three tests which can help you detect COVID, but which do we justify is the best technique? So when you have such kind of confusions, when you perform meta-analysis, you will get a statistically significant value for any particular test. So that will help you to go to the next step, and also to develop a more correct estimate for a significant magnitude. It also provides a complex analysis of harm, safety data, and for other benefits. And it's also used to examine subgroups within individual numbers that are not statistically significant. For example, when we take about again COVID data, when you start analyzing COVID data for the complete the different states in India, it will be a real huge data. So you need to segregate the data into subgroups. For example, different states, different districts, different uh, districts, different taluks. That way, if you start subgrouping it, that it'll reduce the errors, and you can try to pool in the data and try to justify these data as statistically significant. Okay, so how can you basically start differentiating between systematic review from meta analysis? Systematic review is a type of review which determines a question. It sets an inclusion and exclusion criteria for the particular study. We find the literature, we select the literature, and we critically appraise the literature whether, okay, this can be considered for a study or this cannot be considered for a study. And once the literature is synthesized, we have a pooled in literature. So this is just the initial part. But what happens in meta-analysis is nothing but the outcome of systematic review, which is taken, analyzed, statistically processed, and then this synthesis helps you to find the statistical significance, whether we can go ahead, or it will help you to address a research question, whether can we proceed in this direction, is it the right direction, is this the right technique, what I'm conducting for a particular experiment or not. So these kind of questions can be answered by conducting meta-analysis. Right. So how can this meta-analysis be performed? So first, a theory will be postulated based on the research question. Okay, so we go into the database, we try to pull in the research papers from databases such as PubMed, Medline, Google Scholar, or any valid research source of scientific research. And then the first step would be to read through the abstracts of individual paper and check whether it is relevant to our research study or not. Then once we choose, this is the basic way to scrutinize our research papers based on our research interest. Third step would be to find the relevant information from set of these articles, and we extract them into the form of maybe Excel sheets or Word documents for our further steps. Then step 4 would include quantity of the information of the articles. It will be like determined by, you can validate the quality of the research by using certain softwares like grade or like individually manually. When research scientists do, they for at least 3 to 4 reviewers, they sit and analyze the quality and the type of data, and they then decide whether you can go forward with this particular research article. Maybe out of 135 research papers have been scrutinized for your study, out of which maybe one or two you're not satisfied with the results, or it doesn't look genuine enough. That way, it can be manually rejected from our the research, and that can be excluded. Then the heterogeneity of the articles are determined, like how feasible it is to include these articles in our study, or can it be rejected. So this kind of heterogeneity analysis is performed in the step 5. And after that, the summary of the complete effects is being estimated. Like that is, this step 6 comes in picture when you run the codes, the R codes in using the statistical software. You have a certain codes which is written to perform meta-analysis. So once you run that, you can estimate the odds ratio. There are the different models which run at the back end to generate your results. So that is called, there are two types of models, which is called fixed effect model and random effect model. So basically, when you open any research paper pertaining to meta-analysis, what you will be able to see is the results in the form of forest plots and funnel plots. So what are these forest plots and funnel plots, and how do we analyze them? That I'll be discussing in the upcoming slides. Okay, right. So once the forest plot is constructed, we would need to know how to read, how to analyze the data, and what is the outcome of the meta-analysis we have conducted. So that needs to be understood from the forest plots what you have generated. Okay, so the complete results or the outcome of our study will be analyzed using forest plot. But there is something called publication bias. So in step 7, the extent of publication bias in these articles, it says it is determined using funnel plots. So when you run the codes, simultaneously you can generate both forest plots and funnel plots. So the publication bias, how are articles, that is, for example, as I told you, we have out of the 100 articles, we have chosen the 35 articles. So these 35 articles, when you run the code, that is when you run the R code, once you generate the forest plot and the funnel plot, it will show how it is distributed. That is how much of heterogeneity, how much of differences it is having between each other, how it is scattered, whether it is scattered or does it fall inside the funnel plot, how much of variation they have among themselves. So this kind of analysis can be performed using funnel plot. And I will we'll have in the post noon session, we will be generating both funnel plot and forest plot, and I will make you understand how to interpret the results of a funnel plot and a forest plot. So, and the final step is, as it says, is subgroup analysis and meta regression test would be conducted. In this subgroup analysis can be performed, as I told you, when there is huge data and you start stratifying them. Like, for example, this, like I gave an example of about 35 articles. For example, when you start taking COVID data and for different continents, it will be a huge sum of data. So in that case, you need to stratify them according to the continents, according to the countries, according to the different states, different districts, and then again male, female. So the more you stratify the data, the more you subgroup it, it is easier for analysis, easier for interpretation, and it's easier for us to give a very clear picture of how the data is getting analyzed and what exactly will be the outcome of the final meta analysis. Okay, so 8 basic steps of systematic review and meta analysis would be, as I've already discussed earlier, it will be first thing to formulate the research question, define the inclusion and exclusion criteria. For example, in what do you mean by inclusion and exclusion criteria would be, like if I'm going to take the COVID data, I need to include only the data for the month of for example, May 2023. So the articles which belong to April and prior to that should be excluded. So that the earlier articles will fall in the exclusion criteria, and only the data pertaining to the month of May will fall into the inclusion criteria. That way, we will start stratifying our results. Then to develop a search strategy and to locate a study. So to develop a search strategy, meaning which databases do we approach to get our data. Are you going to rely on Google Scholar or PubMed or worldwide website? Like we have for COVID data, we have like WHO has a special website wherein they publish day-to-day COVID infection rates and numbers. So are you going to look into that? So you're going to start you're going to concentrate on one particular database from which you're going to obtain your research articles, the data for your analysis purpose. And then how do you select the studies? So you may have, okay, fine, I'm going to select the studies based on the number of infection, how many infection, if a person has been infected twice, thrice, how many vaccination has he been taken, is it a primary infection, is it a secondary infection. So based on these kind of selection criteria, I'm going to select the number of studies. Once you have selected the number of articles, once you have shortlisted, for example, out of 100, 35 articles you have shortlisted, okay, fine, these are the only studies that I'm going to conduct meta-analysis for. Then you start extracting the data. There is a predetermined data sheets or like data forms wherein you need to enter the data in the respective format. Only then you'll be able to read the data in our program. Okay, so once the data is extracted either manually or there are certain tools which can be used to extract data. Once this is done, you start assessing the quality of data. Like for example, research scientists who are working in this particular field, they'll be well aware of the type of data, the the results what they have obtained. Some, like, once you look into the data, you'll be able to analyze whether it is biased or not biased. So research experts from the certain field who are reviewing the work will be able to assess the quality of data. So once you assess the quality, you analyze the data, you interpret the results, and you disseminate the findings. Okay, finally, I can conclude that in the last 1 month, the number of infections in Bangalore was about 85% when compared to that in some other city, maybe Mumbai, which was about 63%. And then in the other thing, so the prevalence rate is this much. So this way you'll be able to justify, okay, this is the type of COVID infections which is happening in the last 1 month in different cities. Okay, now as I was telling you, we have Prisma guidelines or Prisma checklist for performing systematic reviews. Okay, so the first step of uh, meta-analysis is nothing but the systematic review. For systematic review, as I told you, we have certain inclusion and exclusion criteria. Okay, so what we do, we initially go and search the website. Let me just pick up a pointer. Yeah. So first, when we go and look into the database, we look and we try to pull up some data. Like we get some about 100 to 150 research articles, for example. So initially, we get about 100 articles. So when you prepare a Prisma flow diagram, you need to address all these components. The studies included in the previous version of the review, that was n is equal to 100. Okay, of which the reports of studies included in the previous version of review. So in this, again, we're going to narrow it down. Out of 100, okay, I feel about 75 articles have been they're genuine enough. I can rely on the research results that can be considered based on my inclusion and exclusion criteria. That will be again narrowed down to 75. After that, we are identifying the new type of studies, like number of databases. From databases, how many articles have we got? From the different registers, how many articles have we received? Then how many have we screened? Okay, out of the 75, we have screened about that. For example, in this stage, we have got about 45 articles, of which we are excluding about 10 articles which are not genuine. It is not satisfying my exclusion criteria, of which it will come down to again 35. The final number of total number of studies included in the review finally would be 35 out of 100. So the rest of the numbers which could not be completely retrieved or which are included comes in these blocks here. Okay, similarly, in case if we have identified some new studies, like for example, we have been preparing systematic, I mean, this is quite a long process when you have to compare systematic reviews for about for example, prevalence of brucellosis disease for the past 5 years in India. So till maybe I would have done my literature review till the month of December, after which again when you start preparing your present like your manuscript or something, or when you start preparing your analysis part, you will get to see there are few newly added research articles from the month of January to April. So that also need to be included, which cannot be excluded. You will need to mention how many new articles you have added, from where have you retrieved, does it satisfy your eligibility criteria, and then finally consider it for your meta-analysis studies. Okay, so this the Prisma flow diagram is mentioned on your left hand side. This is how it can be. It can also be modified according to your study as it is given on the right hand side. For example, 12,106 articles were identified. These many were from PubMed, these many were from Scopus, and these many articles were from Web of Sciences, of which some articles did not satisfy the criteria, and they have given an example of robotics and non-robotics here. So out of which 1,873 articles were from robotics approach, and then what it so happens that there are some duplicate articles. Some would be case reports, some would be surgical technique descriptions. So those kind of thing, if you do not want to include for your systematic review, that can be excluded, of which again 900 articles have satisfied my eligibility criteria, that will be further taken for meta-analysis. So for any research paper that you're going to submit to the public repository, you need to perform you need to additionally attach your manuscript along with this Prisma flow diagram and the Prisma checklist when you're going to submit it to any research journal. So this is a prerequisite for any systematic review and meta-analysis paper, research paper. Okay, so this is what I was telling you is a Prisma checklist. So this is available from this Prisma from this website www.prismastatement.org. It is available both in PDF and Word format, in editable format, so that you just need to enter your data here. For example, what is the prerequisite of a systematic review and meta-analysis research paper? So it should have a title which identify the report as a systematic review, meta-analysis, or both. For example, I would say my title would be Analysis of Brucellosis Prevalence in India for the last 5 years in species such as cows and buffaloes. Then it will be followed by a systematic review and meta-analysis, maybe a colon, and then I would mention a systematic review and meta-analysis. If it is only a systematic review, you need to mention a systematic review on. If you're performing only meta-analysis, you can write it as meta-analysis on. So your title should be very specific, and it should state whether it is a systematic review, meta-analysis, or both. If yes, you need to in this last column you have to mention the page number where you're reporting this. That is, it will be usually on page number 1. So to this right column completely gives you the information of in which page what have you stated. Okay. Similarly, the abstract of a systematic review should have the Prisma checklist. That is, according to the Prisma checklist, what all are the information that is to be included in the abstract. It should have a basic introduction, materials and methods. In materials and method, the basic eligibility criteria, that is the inclusion and the exclusion criteria, the information sources, the search strategy, like which other websites, databases, or the filters, the keywords, what were used for searching your the type of literatures that you've used for your analysis process, and on what basis have you done the selection. That is your search strategy, your selection process, for example, what kind of study it was, when was the study conducted. All these kind of details would be included in the selection process. How was the data collected? How was the data item sorted? Okay, have you performed risk of bias assessment? So this is also a prerequisite in any of the meta-analysis. If you perform, it is mandatory for you to address the risk of bias assessment. So that also will be discussing in the upcoming slides. So what is the effect measures, whether it is a risk ratio that you're analyzing? Are you going to analyze the mean? Are you going to analyze the differences? So what is the outcome of your meta-analysis? How are you going to justify? So all these kind of information should be mentioned, and it should be stated in your paper, and on which page number have you addressed. So that should be completely listed here. Similarly, you have to report the bias assessment and certain assessments. So all these information should be addressed in any and every systematic review and meta-analysis papers, and this checklist should be attached to your manuscript for submission. This is a very important prerequisite for any research paper when you conduct systematic review and meta-analysis. So I hope it was a very brief introduction regarding systematic review and meta-analysis. Now I would like to share with you all what are the different softwares that can be used to perform meta-analysis. So there are many online softwares that is available to perform meta-analysis. Revman 5, R software, JASP, Meta Excel, which is an add-on to Excel, but I have not tried to use meta-analysis on Excel so far. I feel very comfortable using R software, R Studio, which is very user friendly. Even if you do not know the basics of software or coding, once you get to know, once you download the codes, you will be able to like as a graduate or a postgraduate, you'll be very well able to make out where to insert your data and how to get the output. So that is done by R Studio, and I really appreciate the person. And this R software is widely used in different biological, biotechnological applications, and it's very user friendly. You do not have to have a you need not be from a coding background or a software background to understand and work on R Studio. So these are the different softwares that can be used to perform meta-analysis. You can go ahead and download, but I specifically recommend you all to use R software or R Studio. And there are different packages that needs to be installed and run to obtain your results of meta-analysis. Okay. Apart from meta-analysis, there are various statistical analysis softwares that is free and online that can be used to for your research purpose. So these are few examples. I can share this link with you all later. Like all the links what I mentioned in my presentation, I'll share it with you all in the second half, so that anytime when you're free, when you find time, you can download, go through these, and apart from Excel and GraphPad Prism, these two are very well known and easy usable software. There are many softwares which helps with your statistical analysis. You just need to enter the data, you get the like results, and some softwares also provide you maybe 1 or 2 line explanation on what exactly you've analyzed. The major drawback or the problem what we as biotech or life science researchers faces, we conduct research, we do statistical analysis, but we are not able to interpret the statistical data. So this kind of like the drawback or setback can be overcome using certain softwares like Skylab, and there are few softwares which will give you the interpretation of like what exactly is the result and what is it interpreting. So this also, these problems also can be solved using some statistical softwares, like free statistical softwares what I've mentioned here. Okay. Now, when we conduct meta-analysis, we come across different types of numerical data. So, what are the different types of data, and how do we analyze, or how do we segregate them, or how do we analyze or arrange them for the purpose of um, conducting meta-analysis? So, for example, a single group study data, they usually have two variables. For example, data and events, total positive, and total. For example, I am going to test, I'll give you an example for a single group study. Suppose I'm going to test about 10 people in my locality or in my area who are COVID positive. Okay, so out of 10, the variables would be total is 10, and of which how many are positive. For example, 3 are positive. So this kind of data is called a single group study data, where there are only two variables, that is the positive, the total positive, and the number of the positive and the total. The total is 10 and the positive is 3. So there are only just two variables. Okay. So this kind of study can be called as a single group study. When you have multiple group study, there are more than three variables. For example, we have mean and SD values, and there are about mean and SD values for about different, for example, mean and SD values for different states and different, like for example, Bangalore, Chennai, Mumbai. Of these, we are analyzing mean and SD data of, for example, some growth rate of some microorganism. So in that you have about different groups, about 5 states, and then individual mean and SD values. So in which then there are many variables, more than 3 variables. That is called, that kind of data can be called as a multiple group study, and it can also be analyzed using meta-analysis. The outcome can be analyzed and you will get a statistical value where you will be able to justify out of these many states, uh, this particular state has the highest mean or highest SD, that way. Now, so you will also come across certain test results or certain research where they have, for example, in case of medical field, you have case control data, clinical trial data, where there are more than 4 to 5 variables. Okay, in that case, odds ratio would it will be given in the form of odds ratio. So those kind of data can also be analyzed. Certain data, like certain research, can give you values in the form of correlation. Some will give you in the form of mean and SD. Some will give you as positive, negative. So these are the different forms of data that can be analyzed using meta-analysis. Okay, so the when you have correlation values, those values can also be analyzed and you will be able to justify your result using meta-analysis. So we have different forms and different types of research data that can be used in meta-analysis. So you need to identify, or you need to uh, like prepare your data in the form where you need to be analyzed and interpreted in a statistical way. Excuse me, just please give me a minute. Well, fine. So here is an example of single group study. Okay, here this data was uh, like generated based on the research articles which we had obtained from the general database. So how I have categorized this data as, the name of the study would be a first table. The column name has, I have segregated as study, events, total. Under the study column, I have listed the names of the author or the article from which I've taken the data. For example, Chakrabarti et al. In detection of brucellosis organism. Okay, this was the name of the study, of which in that particular research he had analyzed 51. The total number of animals that he had tested was about 51, of which the events, that is 30, were positive. So the number of positives have been reported as events, and the total has been reported in the total column. So once you prepare your data in this manner, this is your single group study. Once you run your meta-analysis code, that is our code, what you would get is a result something similar to this. This is nothing but a forest plot of the events, the total, and the different studies. Okay, here, as I told you, as I have spoken earlier, there are two types of models which run at the back end, which is called a fixed effect model and a random effect model. This is an inbuilt model, a statistical model, which runs inside the R program, which will give you the uh, the final outcome of your the meta-analysis is nothing but the percentage. Here we are checking on the percentage prevalence of brucellosis. Okay, so according to the fixed effect model, it says 42% is the overall prevalence of brucellosis by analyzing all these research articles. Okay, so 42% is what is the percentage of prevalence using the fixed effect model, and 41 percentage is nothing but the prevalence using the random effects model. Okay, so this is one kind of data. Okay, as I told you, multiple group data has many variables. That is, they have the number of patients, the duration of, like in mean, the ST value. Then you have some more B cell mean and C cell mean. So this was an example of a multiple group study where they have tabulated many numerical values. So these kind of values also can be used in meta-analysis. There are certain codes which will run mean and ST programs, and you will be able to analyze how significant they are. So out of this, which study is more significant, which has a higher weight, which has a lower weight. Okay, so these analysis also can be performed when the data is a multiple group study. And there are also odds ratio where effect size and variance of certain research has given. For example, when you consider a BCG vaccination trial, T positive and T negative is nothing but the positive group and the negative test group. In that case, you will see group A and group B. Now you segregate your participants or your subjects into group A and group B, where you have about 50 participants in group A and 50 participants in group B. So either they test positive or they test negative after the infection. So though these kind of samples are nothing but they call effect size samples, and they can also be, these kind of data can also be analyzed using our software. Similarly, correlation values. Now, suppose I have a very small test where I've conducted correlation, and these are the correlation values. These many samples were considered, of which I'm going to run the R codes and find out how statistically significant they are by running meta-analysis codes. Okay, so once you run meta-analysis using our software, what are the different types of outputs or outcomes of meta-analysis, and how do you segregate or try to identify what is the results, or how do you interpret the results? So as I showed you earlier, this was from a research paper which analyzed the prevalence of ESBL in India from 2013 to 2019. So it says, according to the fixed effect model, 24% here, what you can see is the prevalence of ESBL in India according to the fixed effect model. So this is the representation of a forest plot. Okay, this is the form of meta-analysis results. Now, publication bias, as I spoke to you earlier, is performed in it also can be generated in our software. Okay, this result is nothing but a funnel plot wherein you can see the number of articles included and how many have outliers. That is, out of, for example, 35 articles we have included, of which about 1 2 3 4 5 articles are not in the same category. That is, they have some differences, they are not similar or they are not heterogeneous. They have their they're not homogeneous, they are heterogeneous. That is, they have some differences, they're not falling into the same category. So I will try to help you to understand how this results can be read and how to interpret this forest plot, or what exactly can you interpret from the forest plot. So these are the two types of output of meta-analysis. Apart from that, there are other plots that also can be generated in meta-analysis. For example, Galbraith plot, LAD plot, Boychart plot, and bubble plot. So each of these plots have their significance, and it all depends on the author or the researcher. How do you want to interpret your data? So any and every research paper, when you read on systematic review or meta-analysis, they would portray forest plot and funnel plot. So that's very easy to understand, and the interpretation of the results is also convenient, and you can well justify using when you interpret your data in the form of forest plot. It is not that they do not represent using Galbraith plot or bubble plot. It is again the wish of a researcher or the author, how he wants to interpret his results. Okay, now forest
Ma'am,
yes ma'am.
excuse me. Yeah, maybe we can just we can take the, since it's a long session, so we can be have some question answers
Yeah,
from the
right now.
participants.
Right.
Yeah, ma'am.
Let's I'll continue maybe after a small Q&A session.
Yeah, because maybe because it's a long
to
stretch.
understand. Right.
Yeah, yes. So participants, you can ask whatever questions and doubts you have till now, so that it makes you also clear, and then we can resume again.
Anyone?
Hi, Sinja.
Hello, sir.
Yeah, great presentation.
Thank
So
you.
I just have one question. Like you mentioned initially that via meta-analysis or systematic review,
Mhm.
we can perform quantitative and qualitative analysis of the data, right?
Right.
So for all the examples or like the part or the paper which I have been through,
Mhm.
like most of them, they like we perform certain sort of analysis over quantitative data, right?
Right.
Whether it is in like prevalence or let's suppose case study or some sort of that design.
Right.
How would you like? Is there any way we can do in the categorical variable or the qualitative variable? Can we do some sort of analysis on that?
So qualitative wise, actually there are certain tests, or I can tell you, like, please give me a minute. Um,
Mhm.
there's a scoring system. For example, I'm just giving you an example. Like, suppose you have some 35 research articles which you need to perform meta-analysis. Okay, so how genuinely can you consider the data for your analysis? There are some scoring system, like Newcastle Ottawa scale, and there are I'm not getting the exact name. Oxen scoring system. I'll just let you know the other name, maybe in the afternoon session. So based on your articles, you can give them the score. Like, for example, how the testing methodology, or the type of test what they've used. So you can give them the score. Okay, so this will give you a quality score for the type of articles that you have considered, which can be taken for your qualitative analysis. Getting my point, sir?
Yeah. Okay. Thank you.
Okay, so you're scoring your articles. Basically, based on the score, you decide the type of the quality of your research articles considered. I'll make a note of this question, and I'll let you know the different types of scoring methodologies that can be used in meta-analysis.
Sure. Thank you. Thank you so
Yes,
much.
sir.
One more question is there in the chat box, ma'am, from Naresh Tare. So he is asking, like, as every study is not does not follow common protocol, and
Right.
result also depend upon how stringently follow these protocols,
Right.
and what associated facilities are available. So collecting different study and then comparison. So how does it correlate? How to avoid the bias
Yeah,
in such
exactly.
cases?
So in this case, we try to analyze the bias in our studies, ma'am. So one is we have to perform the risk of bias assessment and publication bias. So these are the two biases which appears in any meta-analysis procedure. Okay, so this will be I will be discussing regarding this bias in the upcoming presentation, in the upcoming slides.
Okay, so there are strategies also how to
Right.
avoid these biases also,
Yes,
right, ma'am?
yes, ma'am. We need to
Okay,
do the analysis. How do we analyze the risk of bias which has been appeared during the study, or during the period of study, or maybe like during the interpretation of results? So that also needs to be addressed even before you can
yeah.
perform, like
Okay,
even
ma'am.
before you can justify your meta-analysis results.
Okay. So basically, this meta-analysis will be like it is more of the refined output of the systematic review.
Exactly, ma'am.
Okay.
Okay.
Okay. Okay, ma'am. Okay, thank you, ma'am. Any more questions, participants? No?
I had a question on Cochrane review database.
Okay, ma'am.
Yeah, earlier when I saw about meta-analysis, how to do it, if that was the main database from which data was taken. Is that so, or do we still use it?
We do use different databases, ma'am. Like medical data, I think they specify to use Cochrane database.
Yeah,
And
yeah.
we life science researchers, we go by PubMed, Google Scholar. So there is nothing which is not genuine as such, but we prefer the type of articles. We look into most of it which is Scopus index and genuinely published research articles which have
Yeah.
been indexed in high index journals. Like you'll be able to segregate based on like reputed journals and subrated journals, right?
Okay,
So
okay. Thank you.
yeah, based on that we need to do the selection of data.
Okay, thank you.
Any other questions, ma'am?
Okay. So if there are no questions, we can resume back with the next session, ma'am. We can
uh,
continue.
Ma'am, can you give me a 2 minutes break, please?
Sure, sure, sure. Sure,
Yeah,
then. Okay. Yeah, yeah.
yeah, thank you.